Unit 1, Chemistry of Light. The first block is atoms. When atoms form a bond, they release energy, and to break a bond, you have to invest energy. Within a molecule, you have two types of bonds: ionic bonds, where electrons are transferred entirely, and covalent bonds, where electrons are shared equally or unequally. In between molecules, there are three types of bonds: hydrogen bonding, dipole-dipole interactions, and van der Waals forces, and that's in order of decreasing strength. The second block is H bonds. H bonds keep two DNA strands together, and they give water its numerous properties, like cohesion, water binding to water; adhesion, which is water binding to something else; and its other properties like high specific heat, its high heat of vaporization, and the fact that water is like a universal solvent. If you increase the H plus concentration, your pH decreases. The third block is organic compounds, and there are four classes. The first is carbs. They function in energy, structure, and cell signaling, and they're further subdivided into monosaccharides, disaccharides, and polysaccharides. The formula for a carbohydrate is some multiple of CH2O. If you burn 1 gram of carbs, you release 4 calories of energy. The second class is lipids, which is classified into fats, phospholipids, and steroids. Fats are made from one glycerol molecule and 3 fatty acid chains, which can be saturated or unsaturated. Phospholipids are obviously found in the phospholipid bilayer, and steroids are 4 fused carbon rings, example being cholesterol. If you burn 1 gram of fat, you release 9 calories of heat. Lipids function in energy storage, structure, and hormones. The third class is proteins, which are made of amino acids. You have 20 of them. The acidic, negatively charged ones are glutamic acid and aspartic acid, and the basic, positively charged ones are lysine, histidine, and arginine. Proteins have some of the most diverse functionalities, like motility, ATP production, and cell signaling. And the last class is nucleic acids, like DNA and RNA, and they're made up of nucleotides, which are composed of a nitrogenous base, phosphate, and sugar. Unit 2, Cell Structure and Function. The first block is cells. All cells are classified as prokaryotic or eukaryotic. Bacteria and archaea are prokaryotic, while protists, fungi, animal cells, and plant cells are eukaryotic. Second block is cell components. The nucleus stores DNA. The ribosome makes protein. Rough ER modifies proteins. The smooth ER stores calcium ions, detoxifies, and also makes steroids. The Golgi modifies, sorts, and packages proteins, while the lysosome is involved in digestion and autophagy. You have the mitochondria, which produces ATP via oxidative phosphorylation, but also breaks down fats via beta oxidation. You have the cytoskeleton, which has microtubules that move chromosomes, intermediate filaments that make up the nuclear lamina under the nucleus, and the microfilaments like actin, which are involved in muscle contraction and forming the cell cortex under the plasma membrane. And lastly, centrosomes and centrioles, which form microtubules. Plants don't have centrosomes and centrioles, but they do have mitochondria and chloroplasts because they are involved in cellular respiration and photosynthesis. And lastly, prokaryotic cells don't have membrane-bound organelles, but they do have ribosomes because they need to make protein. The third block is osmosis. Osmosis is the movement of water, and keep in mind, water always moves from areas that are hypotonic to areas that are hypertonic. And fourth block is transport. Transport can be active or passive. Forms of passive transport are diffusion and facilitated diffusion, which can be in the form of ion channels or transport proteins. And active transport expends energy. It can be in the form of a proton pump, sodium-potassium pump, or a cotransporter, which uses the gradient of one molecule to drive the active transport of another molecule against its gradient. Unit 3, Cellular Energetics. The first block is energy. Delta G is the difference in free energy between reactants and products. When delta G is negative, the reaction is spontaneous. Enzymes don't change delta G, but they do decrease the energy of activation. The second block is cellular respiration, which is a series of oxidative reactions that break down glucose to form ATP. It consists of 4 different steps. The first is glycolysis, and it splits glucose into 2 pyruvates and also forms some ATP and NADH as well, and this occurs anaerobically. The second is pyruvate being actively transported into the matrix of the mitochondrion, where that 3-carbon pyruvate is oxidized into a 2-carbon acetyl-CoA via pyruvate dehydrogenase, PDH. And this also produces some NADH and CO2 as a byproduct. The third step is that acetyl-CoA then being fed into the Krebs cycle, which will complete the oxidation of glucose. Each turn of that Krebs cycle will completely oxidize 1 molecule of acetyl-CoA, and it will produce some CO2, NADH, FADH2, and 1 molecule of ATP by substrate-level phosphorylation. The fourth and final step is oxidative phosphorylation, which is further split into the electron transport chain and chemiosmosis. In the ETC, electrons are harvested from all those NADHs and FADH2s to pump protons into the intermembrane space of the mitochondrion. And then in chemiosmosis, that proton gradient flows down into the matrix and it's utilized to produce ATP. Ultimately, each molecule of glucose will yield about 26 to 28 molecules of ATP. The third block is fermentation. In anaerobic conditions, the pyruvate at the end of glycolysis can undergo lactic acid or alcohol fermentation in order to produce NAD+, which can be reutilized by glycolysis to produce ATP. And the fourth block is photosynthesis, which is a process by which light energy is utilized to convert CO2 and water into glucose. And it's split into the light and dark reactions. In the light reactions, electrons are harvested from water and funneled through the ETC into NADPH, and this occurs in the thylakoid membrane. Hydrogen ions are also harvested from water and concentrated into the thylakoid space, and when they flow down their gradient into the stroma, it produces ATP. Now, the NADPH and the ATP from the light reactions are used by the dark reaction in the stroma to fix carbon from CO2 into a 3-carbon glucose precursor molecule called G3P. Unit 4, cell communication and cell cycle. The first block is cell communication. Cells can communicate with each other through direct contact, local, or long-distance signaling. Local signaling can be paracrine, autocrine, or synaptic, while long-distance signaling is endocrine, where hormones travel via blood. The second block is cell signaling. Signal transduction is how signals from outside the cells are communicated to the inside of a cell. It's broken down into signal reception, signal transduction, and cellular response. The signal can be a protein. Binding of the signal to the receptor results in conformational change in the intracellular domain of the receptor, which can initiate a signal transduction pathway. Signals can then be amplified by secondary messengers like cyclic AMP, and a final cellular response can be a transcription factor binding to DNA and promoting gene expression. The third block is feedback loops, which can be positive or negative. Negative feedback loops dampen the stimulus. So say your thermostat is 70 degrees Fahrenheit, it gets hot and increases to 73 degrees Fahrenheit. The AC will turn on and cool the room back to 70. That's negative feedback. Positive feedback amplifies the stimulus. An example is childbirth. The pressure of a baby's head on the uterus can increase contractions, which in turn then increase the pressure on the uterus, and vice versa. The next block is cell cycle, which is split into interphase, which is G1, S, G2, and M phase, which is mitosis and cytokinesis. G1 is for cell growth, S is for DNA synthesis, G2 is for more cell growth, while mitosis is the separation of chromosomes, and cytokinesis is the separation of cytoplasm. The next block is mitosis, which is essentially copies a cell. It's split into, prophase, where the chromosomes condense and spindles start to form as the centrosomes move apart. And then in metaphase, chromosomes align at the metaphase plate single file. And then in anaphase, sister chromatids separate. And in telophase, the nuclear envelope reforms, while in cytokinesis, the cytoplasm separates. Next block is checkpoints. There are 3 of them that you should know. The first is a cell growth checkpoint, and this occurs near the end of G1, and it basically makes sure that the cell is large enough. If not, then the cell enters G0. The next cell checkpoint is DNA synthesis checkpoint, which occurs during S phase, and it just ensures that the DNA has been replicated properly. And the third checkpoint is the M checkpoint. And this occurs during mitosis and ensures that the chromosomes are attached to spindles prior to anaphase, because you want to make sure everything separates properly. Last block is cell cycle control, and cyclins and CDKs control the progress through mitosis. Cyclin concentration can fluctuate, but the CDK concentration stays constant. Cyclin starts to increase in late S phase, and it combines with CDK to form MPF. And when enough MPF accumulates, the cell passes a G2 checkpoint and mitosis can begin. Unit 5, hereditary. The first block is meiosis, which forms gametes. It's split into meiosis 1 and meiosis 2. In meiosis 1, homologous chromosomes separate. It looks like 2 X's separating, and in meiosis 2, sister chromatids separate. So a single X separates into 2 lines. The next block is the law of segregation and independent assortment. For the law of segregation, that means alleles for a single gene segregate during gamete formation and end up in different gametes. While the law of independent assortment means that alleles from 2 different genes assort independently during gamete formation. But this shouldn't really apply for linked genes. The third block is Punnett squares. If you see a 3-to-1 ratio, it's a monohybrid cross where the parents are both hybrids. If you see a 1-to-1 ratio, think test cross, where one parent is a hybrid and the other is a pure recessive. A 1-to-2-to-1 ratio, that should be incomplete dominance, where one parent is a pure recessive and another is pure dominant. And then finally, a 9-3-3-1 ratio, think dihybrid cross, where both parents are hybrids for two different genes. The next are some definitions. Polygenic inheritance is multiple genes contributing to a single phenotype, such as skin color, while pleiotropy is a single gene having multiple phenotypic effects. Finally, recombination frequency is high for two genes that are far apart on the same chromosome because the probability of a crossover event occurring between them is higher. Unit 6, Gene Expression and Regulation. The first block is DNA. So DNA is made up from nucleotides, which are composed of a phosphate, a 5-carbon sugar, and a nitrogenous base. The sugar is either a deoxyribose, as in DNA, or a ribose, as in RNA. The nitrogenous base is either a purine, which is adenine or guanine, and you can remember that with the mnemonic pure as gold, or pyrimidines, which is a cytosine, uracil, or a thymine, and you can remember that with the mnemonic cut. Purines are the big ones. A's and T's go together with 2 H bonds, and G's and C's go together with 3 H bonds. The 5' end has the phosphate, and the 3' end has hydroxyl. And remember, DNA only extends at the 3' end. Next block is replication. DNA replication is semi-conservative and it requires a primer. Helicase unwinds the DNA while SSB stabilizes the unwound DNA. Primase will lay down a RNA primer from which DNA polymerase can later initiate replication, and topoisomerase creates nicks in the DNA strand to relieve strain and detangle DNA. Next block is transcription. The central dogma is that DNA gets transcribed into RNA, which then gets translated into protein. For transcription, RNA polymerase does not require a primer, but it does require transcription factors to bind to the promoter in eukaryotes. mRNA is also synthesized 5' to 3'. And when it's processed, you add a 5' guanine cap, you excise the introns, and you add a 3' poly A tail. Next block is amino acids and codons and tRNAs. So you have 20 amino acids. Codons are 3 letters, and there are 4 options per letter. So you have 4 times 4 times 4, so you have 64 codons total. Out of those 64, 3 are stop codons, and you can remember them with the mnemonic: you are annoying, you go away, and you are gone. UAA, UGA, and UAG are the stop codons. The start codon is AUG. There are 20 amino acyl tRNA synthetases, and they recognize different tRNAs, and they attach them to the corresponding amino acid, and it's an ATP-dependent process. The next block is translation. The ribosome has 3 sites: exit, protein, and arrival, EPA. And the existing polypeptide sits on the tRNA in the P site for protein. First step is initiation. The new tRNA arrives at the A site. This requires ATP. Second step is elongation. A peptide bond forms between the C terminus of the peptide and the P site with the new amino acid in the A site. And then the third step is translocation. An ATP is expended to translocate everything. So now the empty tRNA in the P site shifts to the E site and is ejected. And then the elongated peptide on the tRNA in the A site shifts to the P site. Next block is gene regulation. In prokaryotes, genes are regulated with operons, which are composed of a promoter, where RNA polymerase binds, the operator, where regulatory proteins bind, and the third component is the genes being regulated. In eukaryotes, gene expression is more complicated, and it's regulated through various processes like histone modifications, DNA methylation, activator and repressor proteins, mRNA degradation, and protein degradation via ubiquitination. Last block is biotechnology. PCR amplifies DNA. Bacterial transformation introduces foreign DNA into a cell via either a temperature or electric shock. And gel electrophoresis separates molecules based on size and charge. Sanger sequencing sequences DNA by using fluorescently labeled dideoxyribonucleotides that terminate DNA synthesis. You end up with DNA fragments that differ in length and can be separated in a column and then interrogated with a laser that will tell you what dideoxyribonucleotide was last incorporated, either A, T, G, or C, and this will give you the DNA sequence. Unit 7, Natural Selection. The first block is on evolution. Individuals don't evolve, but populations do. Individuals will have inherent genetic diversity. When you apply a selective pressure, certain individuals will survive and pass on their genes, and that's called natural selection. There are three different types. You have stabilizing, which eliminates extremes. You have disruptive, which increases the extreme at the expense of the intermediate form. And then you have directional, where one phenotype replaces the other. You also have artificial selection, in which humans selectively will breed certain strains of plants or animals for particular traits that they're trying to enrich for. The next block is on genetic drift, which is a change in the gene pool due to chance. And there are two types. You have the bottleneck effect, in which a natural disaster, for example, can significantly reduce the population size, and then the resulting population will have a genetic composition different than the original population. And then the second is the founder effect. This is when a small population breaks off of the larger one, and then that new small population has a genetic composition that differs from the original. The next block is on the Hardy-Weinberg equilibrium. This applies for non-evolving populations that are large, isolated, have random mating, and no mutations or natural selection. There are two equations you need to know, p plus q equals 1, and p squared plus 2pq plus q squared equals 1. There are four things you need to know that are very simple, and it's that the p is the frequency of the dominant allele, while q is the frequency of the recessive allele, and that the p squared is the homozygous dominant individual, and q squared is the homozygous recessive individual. Next block is on the evidence for evolution. We get that from the fossil record, which shows transitional fossils, from comparative anatomy, in which we can find homologous structures, from comparative biochemistry, where you can find common metabolic pathways, and also from comparative embryology and molecular biology as well. Next block is on speciation, which can occur from geographic isolation, say from a mountain range, or from polyploidy. If a 2N becomes a 4N, they can no longer mate. It can happen from behavioral isolation, say from different mating behaviors, from temporal isolation, say you have a nocturnal versus a daytime animal, and lastly from reproductive isolation if the two animals simply can't physically mate anymore. And the last block is the Miller-Urey experiment, in which early Earth conditions were simulated by sparking a mixture of water, methane, ammonia, and hydrogen, which resulted in the formation of organic compounds like amino acids. And this suggested a possible mechanism for the origin of life on Earth. Unit 8, Ecology. The first block is population growth, the simplest model of which is exponential, where there is no predation, no parasitism, no competition, no immigration or emigration, and the environment has unlimited resources. But this isn't realistic because any particular area at any particular time will have a limit on the number of individuals that can occupy that, and that is called the carrying capacity. The next block is food webs. Autotrophs are organisms that can produce their own food from sunlight, while heterotrophs are organisms that obtain their energy by consuming other organisms. You have herbivores that feed on plants and carnivores that eat other animals. The next block is symbiosis, which is a prolonged relationship in between two organisms. There are three types: mutualism, which is a symbiotic relationship where both species benefit from the interaction, an example being bees pollinating flowers for nectar and pollen. Then you have parasitism, which involves one organism benefiting at the expense of another, often causing harm to the host. An example is ticks feeding on the blood of mammals. And the third is commensalism, a relationship in which one organism benefits while the other is neither helped nor harmed. And an example is barnacles attaching to whales for transportation without affecting the whale. The last block is diversity. The Simpson's Diversity Index quantifies the biodiversity of a community. It takes into account the number of species present and their relative abundances. The index ranges from 0 to 1, where higher values indicate higher diversity. And that's Unit 8, Ecology. Hey guys, Lutfi here. I just wanted to give a quick message / update. So I was accepted to med school in the US and Australia, and the reason I'm reaching out to you is because of the wild cost of attendance. My US program, which is supposed to start around August, the cost of attendance per annum is estimated to be around 100K per year. And so I'm holding some AP Bio intensive review sessions this Tuesday and Wednesday evenings. I'm going to be covering all the AP Bio content, fast-paced, high-yield content with a bunch of MCQs. If you found this video helpful, and your parents are okay with you investing about $10 to attend a AP Bio review sesh, then go ahead and join one of the sessions below. Even if you can't join, still comment, subscribe, and like. I hope to upload lots of future content to help guide you through everything from taking your AP Bio exam in high school to studying for and content review and acing your pre-med courses in uni to getting accepted to either MD, PhD, or MD PhD programs after uni. I hope you found that helpful, and hope to see you around.